Off-the-shelf, universal CAR T-cell therapies are an emerging approach to chimeric antigen receptor (CAR) T-cell therapy designed to address some of the challenges associated with traditional autologous CAR T treatments. These therapies use allogeneic T-cells from healthy donors, rather than relying on T-cells collected from individual patients, making the process faster, more scalable, and potentially more cost-effective. Advantages of off-the-shelf CAR T-cell therapies. Immediate availability. Traditional CAR T therapies require weeks for the collection, engineering, and expansion of a patient's T-cells. Off-the-shelf CAR T therapies are pre-manufactured and available for immediate use, which is critical for aggressive cancers. Reduced manufacturing costs. A centralized production process enables economies of scale, reducing the per-patient cost compared to individualized manufacturing. Broader patient access. Patients with compromised T-cell function or advanced disease may not be suitable for autologous CAR T therapy. Allogeneic CAR T products are a viable alternative for these patients. Consistency in product quality. Standardized manufacturing processes can ensure uniform quality and potency of the CAR T cells across batches. Challenges of off-the-shelf CAR T therapies. Graft versus host disease, GVHD. Donor T-cells may recognize the patient's tissues as foreign and attack them, leading to GVHD. Solution: gene editing can be used to remove the T-cell receptor, TCR, to prevent GVHD. Host immune rejection. The patient's immune system may recognize the donor cells as foreign and eliminate them, reducing the effectiveness of the therapy. Solution: engineering the cells to reduce immunogenicity, e.g., knocking out HLA molecules, or using transient immunosuppression. Limited persistence. Allogeneic CAR T cells may not persist as long as autologous CAR T cells, which could impact their efficacy. Solution: enhancing the intrinsic survival and expansion capacity of the engineered cells. Technologies enabling off-the-shelf CAR T therapies. Gene editing. Technologies like CRISPR-Cas9, TALENs, or zinc finger nucleases are used to knock out the TCR to prevent GVHD. Knock out HLA molecules to reduce immune rejection. Insert CAR constructs or other genetic modifications. Universal donor cells. Selecting donors with specific HLA profiles or engineering cells to minimize their immunogenicity. Induced pluripotent stem cells, iPSCs. iPSCs can be engineered to produce CAR T cells, enabling a renewable and consistent source of universal CAR T products. Allogeneic natural killer, NK, cells. In some cases, CAR NK cells are used as an alternative to T cells. They inherently have lower GVHD risk and may not require extensive gene editing. Key examples of off-the-shelf CAR T therapies. Allogene therapeutics. Developing universal CAR T therapies for hematologic malignancies using TALEN-based gene editing. Selectis. Pioneering gene editing approaches to create allogeneic CAR T cells. Precision biosciences. Using ARCUS gene editing technology for off-the-shelf CAR T therapies. Fate Therapeutics. Developing iPSC-derived CAR T and CAR NK cell therapies. Future directions. Off-the-shelf CAR T therapies are a promising solution to overcome the logistical and clinical limitations of autologous CAR T treatments. Ongoing research is focusing on improving persistence and efficacy of the cells in the patient, scalability and cost-effectiveness of manufacturing processes, safety through advanced gene editing techniques. These therapies hold the potential to make CAR T treatment more accessible and effective for a wider range of patients.